All right, hello everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me today, also from NCCAT, is Ed Eng. Joining from the Pacific Northwest cryo-EM Center, PNCC, are Kirsten Wheeler and Omar Devolku. And from the Stanford SLAC cryo-EM Center, S2C2, is Michael Schmid. Our speaker today, Vitor Hugo, is joining us from the University of California, Santa Cruz, and Omar is going to introduce him properly after a quick update from each of the 3 centers to keep you up to date on the resources we make available at no cost to the research community. If you do have questions about becoming a user at any of the centers, feel free to send those questions to the panelists at any point during the webinar, or you can send them during the Q&A, and we can address them at the end of the talk. So this seminar is a regular event we host the last Thursday of each month, highlighting methods and tips and tricks that groups can use to collect and interpret the data that they're able to collect using National Center resources. You can save the date already for our talk on June 27th. Kirti Gosapati from Indiana University will be giving a talk on the cryo-EM structure of HIV-1, on the HIV-1 Rev response element RNA and the structural basis of Rev RRE interaction. So a registration link will be up soon on the events page at cryoemcenters.org. Where you can also find recordings of past talks. And one last quick announcement from me. Summer is conference season, and staff from the centers will be at the ACA Structural Science Society meeting in Denver, catching up on all the newest developments in structural biology. On July 7th, we're hosting a practical workshop, Workshop 3, which will include live remote operation of microscopes at the centers from the conference room in Denver. So it will be an intensive one-day dive into cryo-EM with a room full of experts. So a really fantastic opportunity. Registration is still open for that meeting. And I believe poster abstracts are being accepted until tomorrow. So there's still time to get yours in. Then in July 28th to August 1st, you'll find staff from all of the centers at the Microscopy and Microanalysis meeting, M&M, in Cleveland, where we go to both present and catch up on all the cutting edge developments in microscopy. So we hope to see many of you at one of those locations over the summer. Back to today. Please send questions for the speaker at any point using that Q&A box, and Omar will moderate that Q&A at the end of the talk. So let me turn over to our center representatives. Mike, let us know what's new at S2C2.
Good morning. I'm Mike Schmid from the Stanford SLAC cryo-EM Center and from the Stanford cryo-EM Specimen Preparation cryo-ET Center. And we are installing our 4th Creo, which will have all the modern bells and whistles on it, and two other microscopes for screening and training. And for those who are interested in cryo-ET, we are installing a new Hydra to complement our Aquilus for specimen preparation of cryo-ET. It's going to be busy installation summer, but you're all welcome to sign up. One of our popular training and support activities is a weekly session on interpreting CryoSpark Live results for our users, telling them how to interpret their data and how to process it efficiently and pick particles that really are particles. It's a fun session to attend, so please join us for training or screening or specimen data collection. Thank you.
Thank you, Mike. Next, I'll turn over to Ed.
Hi, I'm Ed here at NCCAT in New York. On the data collection front, this summer is going to be a good summer. We're going to have a 5th Krios coming online. On terms of the screening process, We are building our third Glacios, our Glacios Generation 2. Hopefully we'll get that signed off in a month or so. And on the training front, I know summer is conference season, but fall is also conference season. We're hosting 2 workshops, a micro-AD workshop in November and an integrative structural biology workshop in October. If you want to find out more, you can go to nccat.nyspc.org.
Thank you, Ed. And Kirsten, what's up at PNCC?
Let's see, currently at PNCC, we are actually having a Glacios 2 installed. So this will actually help with the 2 proposal types that we have, which is our screening access as well as our general access proposal. Our screening access proposals allow up to 2 people to come on site and prepare their samples and get those ready for additional processing of information that they need. General access proposals are open to data processing as well as screening services, and that is awarded 480 hours of microscope time for 2 years each year. As I said, we've got a Glacios this year, which is being installed right now. It's almost ready for users. However, PNCC has 6 microscopes. We've got the Arctica with a K3 camera for screening and 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. One of our Krios are equipped with Katon, or 3 of our Krios, sorry, are equipped with Katon energy filters, while the other 2 have the Thermo Fisher energy filters. Our Krios 1 just completed its upgrade to a Selectress X, and that also has a Falcon 4i. The newest member is the Glacios 2, however, which we are quite excited to have for screening as well as data collection. If you have interest in actually working with PNCC, you can go ahead and go to pncc.labworks.org and apply today. Deadlines for proposals are the 1st of each month.
All right, so I'm going to stop my sharing and let Omar from PNCC introduce today's speaker.
Thank you, Christina. So it's really a joy to introduce today's speaker, Vitor Serrão, who's the director of the electron microscopy facility at the University of California at Santa Cruz. Vitor has actually been working with all of the national centers. I've been working with them here at PNCC for a number of years now. And he's going to tell us a story in 2 parts. First, describing how an institutional facility can aggregate projects and direct them to work with the national centers. And one of these projects I know is near and dear to Vitor's heart. So it's really exciting to see the progress that's been made. With that, Vitor, why don't you take it away?
Hello, everybody. Thank you so much for this kind of invite. And then I would say that I'm so jealous to hear about all the upgrades that you guys are having. And also, I would like to say that I'm going to be another user for all of you, and then probably keep your schedule quite busy with all the Microsoft and access that you guys offer. So again, thank you so much for this kind of invitation. And then today's talk, we're gonna, as Omar introduced, talking about 2 different perspectives, right? So how a screening facility in a local institution can help a lot the progress of people that doesn't have access to screening microscopes, because the screening facility is quite important to do the hard lifting and then understanding how your sample behaves. And I chose the Selenocysteine Synthase, also called SelA, as an example, because as Omar also said, I have a deep relationship with this molecule that I'm gonna explain along my talk. So starting from the beginning, everything starts once a researcher or a scientist, they have an idea. And sometimes these ideas require some kind of very sophisticated equipment, such as a 300 scope or electron microscopes itself. The screening facilities are here to actually help the scientist and the project go from point A to point B to get access into this equipment. So ideally, we were gonna try to figure out how to prepare your sample in a better fashion to get a suitable grid, beautiful squares, beautiful particles distribution inside the holes, right? But as everybody attending this webinar might know, cryo-EM has a bunch of other advantages in comparison with other methods that I would love to highlight, especially for the second part of my talk. So cryo-EM doesn't require crystallization. It helps you to solve different structures that has multiple conformations, such as the structural flexibility, and you don't have a lot of sample, right? All of these three major topics that I put in here, they are part of the story once I start talking about the selenocysteine synthase, which is you don't require crystallization, or sometimes you have a crystal that never diffracts. You can understand different conformations and sample flexibility, and you can use a very low amount of sample. Okay, about 10 years ago, a little over a decade, the resolution revolution happened, right? So as everybody might know, three major things happens that allows us now to get high resolution reconstruction. So better microscopes, better imaging methods, and more than that, better computers and computational power to actually process these massive amount of data. In terms of sample preparation, we have to also get better and improve. Instead of using our homemade manual plunger, different methods has also been in place to improve your vitrification and avoid a lot of problems that the vitrification process actually can produce. So in terms of the data collection and processing, as everybody knows, we start with a large amount of data, and then we undergo for all the pre-processing, so motion correction, CTF correction, identifying your particles, and then further refine them. So the national facilities are here to help us majority on the image acquisition and then also provide support into the downstream process. But it's also very, very important to optimize this workflow. So the screening facilities helps a lot the sample to be image appropriate. So you go over there with your grid ready to go after you do a bunch of screenings and understanding how your data collection must be done. So I just would like to highlight the importance of having the sample ready to go. So this is the EMDB maps and the PDB models that came from EM maps. In 2022, I would like to give you a highlight over here on the y-axis that everybody can see about 22.5 thousand entries, right? And then only early this week, we can see that this number is reaching way largely. So what happens is the EMDB entries are increasing because the demand is also increasing. But as everybody knows, EM is a pretty expensive method to buy your own microscope and also demands a pretty long data collection. So it's different from like crystal diffraction that you can just put your crystal in front of the synchrotron beam line and get a data set in like milliseconds, a few seconds. Requires at least a few hours of data collection to get a suitable and processable data set. So time and cost are really important for cryo-EM. So this is one of the most critical parts where you should improve your sample before you go into the microscope because the demand is increasing. So the time of getting access to the microscope is also getting harder and harder. And another thing, as I just mentioned, EM is quite expensive. This is a plot talking about how is the market share on the cryo-EM. And a few years ago, it was already $1.2 billion and is expected to reach close to 3.4 in the next 10 years, which means everything is really expensive and must be optimized. So Alex Noble from NYSBC did a great job trying to put it together, all the microscopes around the world. And then this is a beautiful map showing how the microscopes are spread out. And then I would like to highlight the high-end instruments. So the Cryo 300, cryo arms, etc., that actually provide you very high resolution and high throughput fashion. And if we highlight only North America, this is how the microscopes are spread out. But the majority of them belongs to institutes that are either companies or their home institute, like such as a university. And then they should provide service, sometimes pay for service, sometimes it's hard to get access to that unless you start doing collaborations, etc., etc. But NIH started quite a few years ago, and then as you guys just heard about the national centers, these national facilities that actually provide free access for the samples ready to go in order to get high resolution data sets. So those centers are the majority of our usage at the moment in terms of We screen, we do all the hard lifting at UCSC, and then we start spreading out our data acquisition along those centers that we're going to highlight in a little bit. Okay, a screening facility. Everything starts with a user and a drain. So the user walks in your facility with like mice brain extract and have a drain of solving 8 kilodaltons intrinsic disorder protein at atomic resolution, right? So this is everybody's dream, but sometimes doesn't happen, right? And then what we end up is like blobology. We are back 10 years ago. So in order to optimize sample preparation and get a better chance of seeing the holes on your tryptophan, we have to do a lot of optimizations. So this is an ideal grid that if you were lucky, you already had one of these with a very nice eyes spread out, suitable squares for data collection that can last for like a week. And then your particles are well distributed, you don't have preferential orientations, but sometimes this is the reality. So it's very, very common that you have like contaminants, aggregations, your protein likes to stick with the air-water interface. A lot of things can happen. And this takes time to actually improve your sample preparation, your grid preparation to be ready to go for a high resolution data collection. This requires testing of several parameters, such as grid types, if your grid like a different hole size, or if you like gold substrate or carbon substrate, extra film of carbon, how your sample distribution is going to be, the deposition methods, such as Leica, Vitor blot, one-side blotting, double-side blotting, or other many other things, and certainly sample optimization, which requires the sample biochemistry. So it's critical that you have this opportunity to walk in the lab, test different things, screen that right away, because you will never know if your sample is ready to go for high resolution unless you screen. And screening requires microscope time, which, as I mentioned, starts being very, very rare. So ideally, we want the sample distribution to be on the top left corner, but what happens in your initial screen is all the other options. So air-water interface, aggregations, they just like to stick on the carbon, they don't go in the hole, and the work and the hard lifting of a screening facility is actually optimizing those conditions in order to get the best of the best. And another thing that is important, as I just mentioned, screening requires microscope time, right? Because you need to see your particle distribution. You will never know if you have a strong preferential orientation unless you collect a small data set. And these will guide you in order, once you go to high-end instrumentation, to know how we're going to perform our data collection. So, oh, I tested so many different additives and I cannot overcome preferential orientation. So you know by the time that you go into, like, after 6 weeks of waiting list, by the time that you get access to your high-end instrumentation, you have to collect a small data set with untilted, another data set that's tilted, and so on, right? So the screening actually helps you to improve the sample quality, but also helps you to understand your grid. And understanding the grid is the most critical part in order to optimize your data collection and be very, very successful. So we are located, for the ones that don't know or never heard about Santa Cruz, in a Bay Area in the Half Moon Bay. We are located in a beautiful campus in the middle of the mountain with a beautiful sea view that I'm gonna make you jealous at the end of this talk. And we have two different working workflows. One is you provide your sample and you don't want to learn at any point, and I am responsible for doing all the sample preparation, is screening, get access to the high-end instrumentation, and then we go from there for the further data processing later on once we have the massive amount of data. But also we train users, so local users that are getting trained on data processing, and then sample preparation is also part of our services. So, okay, in Santa Cruz, what we have a Glacius 200, which is current coupled to a 10K2 Summit, that we're going to be soon to be replaced by a Thermo Fisher Falcon 4i. This improvement, it will increase our micro ED capabilities and also our high throughput single particle data collection. We do have live data processing, such as CryoSpark Live, Tomo Live, and now we're writing a script to do auto index and integration for micro ED diffraction. In terms of sample preparation, manual plungers, like a GP2, and the favorite of we all, the Vitor Mark IV. And in terms of data management, we are having a 2.5 petabyte storage, the total servers installed in the next 2 months. And we provide remote access to a 13 workstations that the user doesn't actually need to be sitting in there. So you have your own and regular account that you can remote access to one of these workstations and undergo with your data processing. For data acquisition, we have the most common and favorite softwares. For single particle analysis, the same thing. We try to keep updated in terms of all of the current versions of the most used softwares. For cryo-ET, subatomic and everything, same thing. Micro-ED, and then obviously model building and refinement. Everything installed, everything is for free for everybody to get access to it. Okay, this setup actually allows us to have many collaborations and collaborations overseas or at least abroad US. So highlighting the current users that I have in Canada. So from my former institution at University of Toronto, all the numbers that are listed in there, they are the proposals IDs that I have in national facilities. So most of them are from PNCC currently. So from University of Toronto, all of these, from Queen's University to other proposals, and also from Brazil, University of São Paulo, I do have 2 of these proposals. One of them is the main description of this talk. And then from US, we do have a deep interaction with Brandeis University, Stanford, and University of Michigan, and obviously University of Santa Cruz with all of these proposals that are ongoing at PNCC. So talking about some of the results that are not part of the national facility, but that were prepared, screened, ready to go, and then the data collection was done in a high-end instrumentation. Starting with some of the results that we have from UCSF, we have from viral glycoprotein interacting with antibodies, small proteins that are like less than 100 kilodaltons, and such a different ways of doing the data collection in terms of tilted, untilted, etc. And we have these 4 major projects that the data was collected over there, and they're getting ready to be published. And then I would like to take this opportunity to thank David Buckley, because he's very helpful in terms of getting all of these data sets as good as we could get from there. We have been using also University of California, Berkeley, from like large stuff such as ribosomes, and also very small eukaryotic elongation factors that we have been using in those microscopes. And from Janelia, we recently, just a few weeks ago, collected a less than 100 kilodaltons complex, also remotely accessed into their scope. And then we are undergoing for all the data processing. Okay, talking about the NIH-funded national facilities, we have the list of all of these institutions, and I can say that I'm proudly using the majority of them. So Boulder, you are on my list. I'm gonna get into that soon. And I would like to highlight some of the results that we have from these national facilities. From NCAT, so we were just chatting right before this talk. Tellini was a PhD student at Professor Rubin's lab, and she was the first in-person user to go to New York and collect this beautiful nucleus unbound complex that we are finishing the data processing. From S2C2 or SLAC, Professor Harry Noller has been collecting very high resolution ribosomes complex, and all of the samples have been screening and prepared in here. And for medicine, we just prepared using the Leica GP2 these bacterial algae symbioses that will undergo to cryo-ET data collection. And in terms of PNCC, as I mentioned before, we have several proposals that involves from ribosomes, very large molecules, circadian machinery with high symmetry, and very small enzymes in different conformational states with like bound and unbound of the substrates. We have amyloid fibers. We have more viral glycoprotein with different antibodies, with different symmetries and different floppiness, also different virus glycoproteins. And from some of my external users, we have molecular chaperones and all of these structural plasticity of the particles actually swinging around. Some degradase complexes, class 2 fusiogens, the human calpain trees, a collaboration, long-term collaboration with Professor Davis, and now the record of our lab, which is a 52 kilodaltons protein that has been collected a few months ago, and then we're still undergoing into the data processing. So saying that, we have been very successful in these 3 years that we have been fully operational. And this is today's story. So the bacterial Selenocysteine Synthase, a structure at 2.69 angstroms, it does seem like a very easy structure to get, but the journey that I would like to share with you today, it started in 2005, long time ago, even before I joined my undergrad program. And I was gonna try to explain this journey with like all the plot twists and the very, very unique features of this molecule. So we recently published this structure. It's a long-term collaboration with Professor Otávio Timan. But before deep into the structure, I would like to give a brief introduction about selenium and its importance. It was discovered by Berzelius a long time ago, and it is essential to human nutrition. And the selenium deficiency is associated with some neurological disease, such as Alzheimer and Parkinson's. But the major disease that the selenium deficiency can cause is called Kishan disease. It's endemic disease in China because the soil is very, very poor in selenium concentration. But the model organism for today's talk is actually E. coli. And in terms of E. coli, it's important because selenium is responsible for the respiratory chain under anaerobic conditions. But more importantly, a bacteria can survive in a very hostile environment, right? So bacteria can survive if you put a bacteria under high selenium concentrations. And the selenium metabolism is quite important to understand how the bacteria can survive this kind of environments. So one of the mechanisms that bacteria can use to avoid the high selenium concentration inside its environment is actually transforming selenium compounds into selenocysteine, which is not dangerous, but actually helps the biochemistry enzymatic functionalities of the cell. So it was discovered in late 70s, but only the entire biosynthesis was actually fully understood in early 90s. And it requires a very specific machinery involving 5 different enzymes and 2 RNase elements. So 1 tRNA, which is specific for selenocysteine incorporation, and 1 hairpin into the mRNA that actually changes the codon to be not recognized as a stop codon, but actually a selenocysteine incorporation codon. So the biosynthesis of SEC or selenocysteine starts with a serine synthesis using a serial tRNA synthetase and incorporating the serine residue into a SelC or tRNA sec tRNA. And then the conversion from serine to selenocysteine actually happens in a co-translationally way that I'm gonna highlight right now. So as I just mentioned, there is no codon for selenocysteine if you take a look on the 3 letters codon table, but you can actually get a UGA and change this meaning by changing the mRNA structure. And then the biosynthesis of selenocysteine start with serine that you remove the hydroxyl group, and then you incorporate the selenium compounds. In bacteria, this requires these 5 elements. So the serial tRNA synthetase that I just mentioned, responsible to charge the tRNA, and then this big homodecameric purple complex, which is called selenocysteine synthase, or SelA, that we're gonna highlight in the next slides. So the selenium compound is delivered by 2 different enzymes, CSDB and SelD, that are responsible to reduce the selenium concentrations and save the environment from toxicity, and incorporate and deliver those compounds into the SelA. And finally, another specific elongation factor, which is called SelB, is responsible to deliver the tRNA, the amino acylate, the tRNA into the A site of the ribosome. Once we look at the tRNA structure, it's important to highlight that is a little different from the others. First of all, the anticodon is different, but more than that, there's a very long variable arm. And then some of these structures are important that are going to highlight how this is critical to understand that you are not incorporating a serine, but you are actually incorporating a selenocysteine. As I mentioned, the hairpin into the mRNA is called SESIS element or selenocysteine insertion sequence and forms this secondary structure element to modify the UGA codon meaning. Comparing bacteria to other life domains, for example, once we look at the archaea, what happens is if we take the gene or the protein that codifies SelA, there is a unique N-terminal domain in bacteria, and this domain doesn't exist once we look at across archaea sequences. A structure was solved in 2005 by crystallography, and it shows up as a dimer, so it doesn't homodecomerize. So, and this guy doesn't actually present any function. So once we look at the other elements, the tRNA is very similar, the mRNA hairpin is also very similar, and once we look at the structures, we're supposed to be seeing active site. So there's a PLP or a vitamin B6 that I'm going to highlight in the next slides on the binding site. However, there's no function. But the N-terminal seems to be very, very critical to keep selenocysteine functional across the life domains. Once we look, for example, in eukarya, SelA doesn't exist at all. And its function was replaced by two different other enzymes. One that's called PSTK, which is responsible to recognize the tRNA and phosphorylate the serine, and another one that's called SEP-S or SEP, serine phosphorylated serine to SEC, selenocysteine synthase, responsible to perform the second step, which means this is way less efficient in terms of now you don't have a single structure that does the entire entire conversion, but now you split up in two different enzymes. However, if we look at the tRNA and the mRNA, both structures remain intact. So there's something in terms of evolution that changes from bacteria to eukarya and from bacteria to archaea to eukarya. Okay, talking about selenocysteine synthase itself, as I mentioned, is responsible to do this conversion, is called a pentamer of dimers because, as I just showed, if you truncate the N-terminal, becomes a dimer, so you can understand that the entire donut shape, the ring, always shows up once you do have the N-terminal interactions forming this pentamer of dimers, requires PLP to be bound, covalently bound into the binding sites. So it's a PLP-dependent enzyme. The entire complex has about half megadalton, so it's quite large. And it was believed that binds 5 tRNA sec. And this started in 1992 with this incredible work done by Bach et al., where they have done a lot of negative staining with bound and unbound states. And by these class sums at that time, so everybody can pay attention that this started in the early 90s with EM already, you can actually see how the ring conformation it is in there and how it started being fulfilled with all the little spaces, right? So if we compare how dense the electronic dense is dense in some of the regions, they do believe that the proper stoichiometry of one SelA homodecameric state, it is to bind 5 tRNA secs. So one SelA to 5 tRNAs. And this was what was believed for several years. So you might be asking, okay, so it should be easy to get a structure, right? So you know how to express as a bacteria sample, pretty large, easy to put on the grid. So high symmetry, you should be straightforward to like express, purify, put on the grid, and solve the structure right away. But what I'm about to tell you is 20 years of story that requires a lot of screening, a lot of sample preparation, and a lot of optimization. Everything started once Professor Otávio Timan at University of São Paulo, Brazil, started doing selenocysteine studies. So he tried to understand how SelA behaves and works across different species, especially E. coli. And then he had a brilliant Ph.D. student, Dr. Alexandre Casago, that was responsible to understand and solve its structure. So 20 years ago, the method that we have to get high-resolution structures was crystallography. So crystallography has a lot of issues, right? And then one of the things is Sometimes you believe that, okay, if I don't get a crystal, it's my problem. Or what happens once you get so many different crystals, but no one of them diffracts? This becomes a problem, right, to solve the structure. So at the end, we do have a sample that actually crystallizes very nicely, but never diffracts well. And I'm gonna try to show you and hopefully convince you all of the reasons why we never saw any spot from those crystals. Okay, more or less at the same time, a few years later, another brilliant Ph.D. student started in the lab, Dr. Livian Manzini, and she started doing a lot of the biophysical characterization. And then I started working with SelA, but at the same time with her to also try to help to solve its structures. So Dr. Manzini, she figured out how the selenocysteine synthase can be expressed free of endogenous tRNA. And also she showed how the homodecamerization happens. So how one monomer that is expressed actually forms a dimer, the dimer a tetramer, and then so on, right? But the interesting thing that she found is as soon as you start increasing SelA concentrations, SelA start forming these side-by-side and stack conformations, right? So this is really, really tricky to actually get a pure sample that diffracts, because now you might have different conformations to form your crystals, but because they are floppy, they are not touching in a perfect position, you don't have Brock's law being applied. So you don't see spots, but you can actually see structures that are a little random, so they don't diffract well, but they can form a crystal. And this is concentration dependent, which means in order to get a crystal, you have to increase the concentration. So makes everything harder, right? At the same time, she was also the first one to map which one of the structures the SelA recognized on the tRNA. So it's important to highlight that the tRNA sac is unique for selenocysteine, which means each one of these structures can be understood as a checkpoint for the following enzyme. So she proved that SelA, she was the first one showing that SelA only recognizes the acceptor and the D-sem loop. Okay, so this is what we have from early 90s, right? So SelA, class sums, it shouldn't be hard to do, yeah. But one of the things is how many tRNAs are actually bound to this molecule. It was believed to be 5 tRNAs into a homodecamer, but her in solution studies, once she got the endogenous free sample, actually showed 1 to 10. So 10 SelAs binding to 10 tRNAs. So one homodecamer binding to 10 tRNAs. And then we said, okay, we have a problem, right? Because in solution studies are showing things that doesn't match what they reported a few years ago by YAM. So the idea was, okay, why don't we just repeat and do YAM and try to figure out not only looking into the conservations of the densities, but using more sophisticated analysis, right? So we do have all the 5 subunits of SelA that were really canonic, and I hope you can identify that we have a 5-fold symmetry X, right? So it's a D5 symmetry. And this, once you bind to tRNA, might be broken if you have 5 tRNAs, right? Because a tRNA is intrinsically not symmetrical, which means any even number, any odd number that binds to the binding site of SelA that is not like 1 to 5, for example, it might break the symmetry. But once we look at the symmetry around, the symmetry was preserved. And then after aligning those particles, we can actually count 10 of the dots, which means the symmetry actually now agreed what was previously reported by in solution studies of having 1 SelA to 10 tRNA sacs, using exactly the same method as before, just doing a little different analysis. So this is the results that we got like 10 years ago. We got excited, negative stain, everything looks good. We have a sample ready to go. So we have the perfect symmetry. So now it's just go for cryo, right? So resolution revolution was happening. So we prepared cryogrids. We collect the data, we start processing, and I got my perfect blobology map in about, I don't know, probably 20 angstroms. That fits what we were expecting. And then after that, we start processing with the tRNA. We can count 10 tRNAs. Everything was great. But this is the sad plot twist of this talk. There was a science paper, and our name wasn't on it. So Professor Shigeyuki Okoyama from Japan, he was able to crystallize and solve SelA structure, but not from E. coli, from another bacteria called Equifax aeolicus. And it is important to highlight this name and where these bacteria lives, right? So, but before going to that, it's important to highlight that's not only SelA structure that was solved, he also solved the SelA bound to 10 tRNAs and showing everything that we believed, that the D arm is responsible for the specific interaction with the N terminal, and then the acceptor arm is inside the binding pocket. Okay, beautiful. So let's see what this structure can show us. The catalytic mechanism of SelA was always proposed to be a shift-based mechanism where you have like the electrons passing through. But now with the structure, we can actually see this very, very unique arginine pocket that creates a perfect environment for the electrons to flow and do the SN2 attack, and then replace the hydroxyl and incorporate the seleno compounds into there. But then you should be asking me, okay, use of the structure right after from E. coli that you just published, and it's not a science paper, by the way, but why you want to solve it, right? It's just a matter of, yeah, I have my long time story with that. It's a matter of proof that I can do it. But no, it's actually, once you think about Equifax aeolicus, is a bacteria that lives under extreme conditions, right? So extreme, when I say it, is like high temperatures, high pressure, and also high selenium concentration environment. Once we look at the regular E. coli doesn't live in so extreme conditions. So we might see some differences between what was showed before by the crystal structures at like 3.8 angstroms and what we can see right now, specifically once we look at the binding site and understand the mechanism. Okay, so we start again this collaboration years after I finished my PhD to actually solve the high resolution structure. We Professor Siemens sent me the plasmids, I expressed, purified, did a bunch of screenings. As you can remember, is a kind of tricky conditions because I cannot increase the concentrations that much because the SelA start sticking to each other. So I don't want to solve the stacks conformation. I want to solve the free particles. So getting the perfect condition in terms of what is the grid type, what is the concentration, etc., was a little tricky. That requires a lot of the sample optimization. We wrote a proposal, we got accepted. We used the Krios G3Y with a GATN bioquantum K3. And I would like to make a strong point about this because the setup of the data collection actually may change your conclusions and how you can actually see better or worse depending what you are looking for. Okay, so using the GATN bioquantum K3, we got this beautiful roughly 2.7 overall reconstruction with a solid 2 angstroms in the core once we look at the local resolution. As you can also appreciate, SelA has this particular donut or ring shape once you look from the top view, but on the side, it's really, really tricky to do the particle picking. So during the particle picking, you can actually do a ring block picker to identify where the donut shape is, and then another type of picking to identify where the side views are, and then you can combine both of the data sets later on, right? So this is what I did to actually identify the two different sets of particles, combine them together, and get this beautiful structure. Just briefly showing that is a full type 1 PLP dependent enzyme, as several other PLP dependent enzymes mainly reported to be part of the selenocysteine biosynthesis. The topology is quite canonical, and then you can compare with several others, SEPSISA, CBL, other enzymes that are related to either having a PLP bound to the active site or related to the SEC biosynthesis. Okay, talking about the PLP, now we do have a very high resolution around the active site, so we can see the PLP in details. So if we take what is called the canonical dimer, this canonical dimer actually forms the PLP binding pocket, not between these two subunits, but the surrounding subunits. So as you can see over here, hopefully you guys can see my cursor, this is the two intimate dimers, but the binding pocket, which is this blue area over here that's positively charged to bind to the tRNA, is actually formed by the sequential subunits, the sequential dimer. So if we compare how these subunits interact with side by side, we start understanding that the functionality of SelA only happens once you do have the dimer, or at least a tetramer, right? Because the dimer itself forms this very strong interaction in here, but doesn't have the active site, because the active site is just formed between the two surrounded subunits. So once we look at the active site with the PLP, as I mentioned before, we have pretty close 2 angstroms reconstruction, and then we can measure all the atoms distance and recognize where the side chains are pointing towards. So comparing the crystal structure from Equifax, Aeolicus, and the E. coli SelA, and the 2 different resolutions, look at this plot, it's quite hard to tell any differences. But once we actually start identifying, we can see how the arginine from the second subunit is important to actually form this pocket. There's a lot of conservation between the residues, but comparing the Equifax Aeolicus structure, there is this unique serine, right? So as I mentioned before, this type of a bacteria lives in a very high selenium environment, which requires a very fast turnover. So the serine helps to create a better environment for this electron transition inside the binding pocket. So we don't have any serine in any one of the other bacteria that doesn't live in this type of extreme conditions, which is a very unique system to actually be very responsive once the selenium concentration inside the cell environment starts raising up and increase by the any kind of danger metrics, and then you can easily incorporate them into selenocysteine and then survive. Okay, and I would like to also highlight the N-terminal importance, right? So it was proposed that the N-terminal is not only responsible to form the homodecamer, but also to recognize the D arm of the tRNA. So the Professor Shigeyuki Yokoyama group in Japan, they also proved that. So if you truncate the N-terminal and they solve the dimer structure by crystallography, shows that it doesn't have function and doesn't homodecamerize, right? Okay, once we compare our structure with the crystallographic structure, we can clearly see that the N-terminal is missing, right? So why and how can we overcome that? The crystal structure probably the crystal lattice fixed the N-terminal in a particular condition that it was able to be seen, and then phase and solve that particular portion of the molecule. But in EM, things are floppy, and things sometimes they don't align well once you're doing the data processing, right? And then another thing that is important to mention, that the N-terminal is actually composed by 3 alpha helixes. But if you truncate alpha 1, the first portion of the N-terminal, you stop the cell wave function, but you don't stop the homodecamerization, which means it's not the entireness of the N-terminal that is important for the homodecamer assembly, but only the first 9 amino acids that are critical for the tRNA D arm recognition. And this is also another paper that we published years ago, where it shows that not only the tRNA recognition, but also the other enzymes partners recognition. So it completely stops the cell waste function once you truncate only the first portion of the N-terminal. Okay, so saying that, how can we overcome this problem, and how can we start seeing that flop any terminal? So this is the structure that we got. E. coli, SelA, very nice resolution, etc. But talking with Omar he was the Spock for this proposal, we were able to get access for the newly installed Falcon 4i selectors, which actually provide a very, very low dose in terms of what we were collecting. And then we decided to change the strategy a little bit. In terms of particle alignments, particle distribution, sample preparation, it was precisely the same, but the data collection itself changed. And now we were able to start mapping where the N-terminal domain is pointing towards. And once we compare with the crystal structure, we can clearly see that has like floppiness. There is like difference in orientation that the crystal lattice produce and what we were seeing, yeah. And then as I mentioned on my, I think, first or second slide, is like a story that requires 3 different steps. So no cryo-EM helps in stories that doesn't require crystallization, as you guys saw. I wasn't able to get any spot to be diffracted. In the crystal to be diffracted, requires a very low sample concentration because I cannot increase the concentration of these particles, this sample, because they start forming other kind of supramolecular assemblies. And you can understand flexibility. So now we can see the N-terminals where it's pointed towards, and there's a bunch of new questions that open up. Right, so some unsolved questions are, this is the tRNA complex solved at 7.5 angstroms, but it does show all the binding sites completely full. Once we do have EM images, we can see some of them in there and all the binding sites and the previous structure that I solved. But after classifying a lot, you can see some of the sites that are unique bound and some of the others are completely free. So is a very clear non-competitive or sorry, non-cooperative binding method. And this is important to understand why this molecule requires to be an entire homodecamer, but actually doesn't use all the binding sites. Another important question is, as I mentioned before, the tRNA is the checkpoint for all of these interactions, which means you can understand that, okay, at some point, the SelA must interact with the tRNA synthetase bound to the tRNA sac, and then specifically deliver the tRNA into the binding site. So these are still ongoing as the same fashion as the SelD, which is the enzyme responsible for selenium delivery, specifically into the SelA binding site, how this ternary complex looks like. And then Niels Fischer from Professor Marina Rodinina's lab with Holger Stark, they did an incredible cryo-EM work on understanding how cell B specifically delivers the tRNA sac into the A site of the ribosome. However, nobody never understood how the elongation factor can understand and recognize the tRNA sac from cell A. So those are all the remaining questions that we're still working on in collaboration with obviously PNCC and also Professor Timan. With that, I would like to start all of my acknowledgements. First, all of the researchers that helped me to solve this very beautiful structure that thanks for all these nice artistic plots nowadays. We can share some like nice features of those. So Dr. Humberto Pereira, Dr. Karin Minari, and obviously Professor Otávio Timan. I would like to take a special thanks to all of my users and collaborators, first for trusting the UCSC biomolecular cryo-EM facility and become our users, but more than that, for trusting me to actually taking care of your sample preparation optimization. And obviously, I would like to thank all of the national facilities for supporting our users and research, in special to PNCC, PNNL, everybody involved. Irina, I don't know if you are watching this. I promise I'm gonna take it out all my terabytes from there at some point. And I can say that I had the pleasure to work with pretty much every single Spock from PNCC and each one of the data collection and screening sessions. They are incredible and I have been learning a lot. And finally, I would like to thank my supervisors, Professor Melissa Jurica and Seth Rubbing, and all of the labs, research PIs, and groups that have been trusting me over the course of this almost 3 years to help speed up cryo-EM projects and get all of these beautiful structures that I presented today and hopefully to be soon published. And a special thanks to Eric Schell, is our assistant analyst, and all of the data management, workstation setup, all the software upgrades. None of them would be possible without him. He's very, very helpful for us. And then the facility, always I call the facility us, which is me and him. And then we're trying to do our best to keep everything running 24/7. Thanks to NIH for the funds to get the instrument. Please follow us on the Twitter, now X, and Instagram. And if you have any questions, please don't hesitate to follow up on the website and also reach by email. With that, I will take any further questions.
Thank you, Vitor. That was a great talk. It looks like you've stunned the crowd into silence. I don't see any questions right now, but hopefully some will come trickling in. I had one question for you.
Sure.
Is this specific for selenium?
Oh, this machinery, yes.
Yeah. I know some of the
So-
other metals might not be natural, but if we were talking about cobalt phosphate instead of selenophosphate, would that work?
That's a good question. So we know by the standard amino acids table, we should have 20, which are the standard ones. But we have, for example, in bacteria, another 3. So selenocysteine, pyrolysin, and methyl something that I forgot the name. And then these 3 requires a specific biosynthesis path to actually incorporate different things, right? So in terms of method, selenium is unique. So we don't have any other method to be incorporated, at least to be part of a new realm of, for example, enzymes, right? So forming selenoproteins or pyrolysin proteins is something that bacteria and eukaryotes, and archaeas, they done a lot to actually survive this kind of environments, but they don't actually perform any other method. Maybe it might have like some other metal that in high concentrations is very, very toxic, and it might be like some kind of mechanism to survive into those kind of environments.
So for my education, are there a lot of selenoproteins, and in these proteins, do these act as like reduction centers?
Yeah, so it's a very interesting question. So there is like the selenoproteomics studies. So to date, I do believe in humans are found 48 selenoproteins with different locations, so transmembrane proteins, enzymes, soluble proteins, etc. And they do have like different functions. The incorporation in eukarya is a little different because you don't actually die by like going to a selenium environment, so doesn't need to be as efficient as in bacteria, for example. In bacteria, I can recall from the top of my head, but some of the enzymes that are part of the selenocysteine synthesis, actually they are selenoproteins, right? So the SelD that I mentioned before, they do have a selenocysteine in the active site. So it is kind of a feedback loop of using a high concentration of selenium to produce selenocysteine, and then you go over there to produce more SelD that actually captures the selenium from the environment and put on selenocysteine, right? So it's a kind of very interesting loop.
I'm gonna butt in for a second, just in case some of you have meetings at the hour and have to head off. We can stick around and answer a couple more questions if Vitor is able to stay. But for those of you who aren't, we will put the recording up, and there will be a link through that cryoemcenters.org site. And thank you for joining us today. We hope we see you again, either here on Zoom or in person at some point this summer. And I'll let Omar go back to finish up the questions.
So we've got a question that just came in from one of the attendees. You spoke about the missing N-terminal density that you were able to visualize. Was it a change in the processing workflow or the data collection parameters that allowed you to visualize that? And can you discuss what the differences were?
Sure. It is quite a very interesting question because we used exactly the same grids that were previously stored in at PNCC. So same sample, same grid conditions, same data processing workflow. The major difference that I can say, it was the equipment that we use and the data collection parameters. So usually once you start the data collection, you can see your particles, you can see like, okay, I have good contrast, so let's go for the magic number of 50 electrons per angstrom square. Let's do this like that fashion. And then you trying to get a SOP and play by the books because it works for the majority of the samples. But some of the samples, they are a little trickier, right? So if you work with something that's small, you cannot go for that number. You have to reduce your dose. You have to find another way to actually avoid your sample to shake a lot, etc., etc. If you're working with a ribosome, you can pretty much burn your sample because it won't shake that much, right? So the N terminal, it was well known to be floppy, and we couldn't see, right? And one of the reasons why the crystal structure was able to see is because part of the cell-wide structure was actually docking the N terminal and made it be there, right? And this might explain why we couldn't never get any lucky with the crystals that diffract, right? So it's just because too floppy. Knowing that, one of the things that we discussed is like, let's collect in a lower dose with this better setup, collect way more. So we increase the number of movies, we increase the number of particles with a very, very low dose. We kept exactly the same magnification, we kept the same other optics setup, but the only thing that we changed was actually the dose and how many movies we collect. So we changed it a little bit because the Falcon 4i produce EER. So you collect like 320 frames per second. You can pretty much smooth your dose across a lot of frames. So it is very, very helpful once you try to avoid this kind of small movements that at the end, because we have to average a lot. It's easy to average once you have the donut, but things that are slightly floppy like this, even though we are not seeing atomic resolution, but we start seeing those, is a matter of disturbing your sample less. So now we are collecting way better, I would say, in terms of getting more data with lower dose. And then we were able to start seeing the guys that are supposed to be unseeable, right? So it's just because they were floppy before.
Actually, can I ask a quick follow-up? So the number of particles going into your final reconstructions, is that similar? Because you said you have now more data. So is that giving you more particles or more particles that are the same?
I can't remember from the top of my head, but for the final reconstruction that we published, it's about 80,000. So it's not a huge number. And then now we are close to like 250. So increases the number of particles because once we do the 2D classification, we can actually see clearly where the N terminal is, right? So even once you look at the side view, sorry, you're supposed to be seeing like the pinwheels sticking around, but it's hard to see. And we start seeing once you go further on the 3D data processing. And then with that, we're doing a lot of like CTF refinement after get your perfect alignments, etc., etc. We start busting the signal. So indeed, increasing the number of particles helps, but I do believe that actually collecting with a very, very low dose and shaking the sample less actually improve the quality of the data in order to see the N terminal.
I have two quick questions, comments and questions. If these seleno enzymes that are involved in the metabolism, or in fact do incorporate selenium, would these be, and especially since generally speaking, you're saying the bacteria treat things differently from eukaryotes, would these be great drug targets for antibiotics that might interfere with metabolism and thus the survival of bacteria, versus, obviously they would not be targeting eukaryotes then?
Well, it is an interesting point. However, you can knock out all of the selenium machinery from the bacteria and still survive.
Yeah.
Which means it doesn't survive under oxidative stress. right? So unless you get the patient and put under high concentrations of selenium, the bacteria will survive, right? Because at the
That doesn't
end,
sound therapeutic.
exactly. So it's not vital for the bacteria to be
Okay.
alive unless you are in that particular condition.
Okay. Well, one more point is that Alexander Carago was in fact Casago, excuse me, is now working with us, and so
Yeah, I know.
we recognize his value and his accomplishments.
Indeed. Yeah, and then he has done a great job with trying so many crystallization methods. So he tried like in situ proteolysis, he tried different constructs, he tried several things, and then unfortunately never tracked. And by the time that he was leaving the lab, I was joined into this project. So he led me to keep playing with the crystallography aspects of that. I tried several times and I couldn't get any single spot as well. And then that was the time that's like, okay, this thing is not working, so let's change the methods. So we start doing cryo-EM. Actually, it was negative staining at the beginning. So the majority of the first two papers, they came from like negative stained samples. And then after that, understanding like how many tRNAs, how SelA binds. The second part of my PhD degree was actually trying to do the cryo-EM structures of that. And this is the reason why I'm here today, right? So I became the director of the cryo-EM facility because I started doing cryo-EM with something that I couldn't diffract. So SelA at the end is responsible for me to keep running the facility nowadays.
And that's why Alexander is working with us with cryo-EM, right?
Yeah, exactly. So you can see that this enzyme is so important for the field.
Do you think there's any hope that the crystals would diffract electrons better than X-rays?
No idea. I know the diffraction that they got, they were able to phase and solve the structure. The APO SelA, it was a 3.8 pattern, and the tRNA bound was actually 7.5. That they could see where the tRNA is actually pointing out, but as you guys may know, building something into like worse than 5-6 angstroms is like awful. And then this is why I think improving the resolution, not only for the binary complex, the tRNA bound to SelA is important to understand how this flopness is happening, how the active sites are there, etc. It is also important to understand how the mechanism actually works, right? So because clearly we don't need the 10 binding sites to be fulfilled to do the function. Right, this is one thing. But diffracting electrons, this is a good question. I never tried that. So I tried like 2000 crystals. I tried so many different synchrotron beam lines. Dr. Humberto Pereira and me, we pretty much shipped SelA crystals to every single high-end crystallization or synchrotron facility around the world. So those crystals, they traveled a lot.
All right, well, thank you for sticking around an extra couple of minutes to get to all those questions.
Sure, it was a pleasure. Thank you so much for the invitation again.
Yeah, thank you for a great talk.
Yeah, thanks.